There we go.
Right. Hi, everyone. Hi, Mom and Dad. Welcome to Get Species Richard by Tryin, the newest installment in the UBC Backyard Naturalist series. We're going to be talking about biodiversity today, featuring 50 Cent. I'm Clare Eigenbrode, and I'm joined today by Lindsey Bereckis.
Okay, so I guess Claire already kind of introduced me, but I could talk a little bit about my academic background and my interests. So, hi everyone, I'm Lindsey Bereckis. I got my bachelor's in zoology and a co-major of sustainability from Miami University. I graduated in 2021. It seems like forever ago. But I'm interested in urban ecology, sustainability, and geographic information systems, or GIS. And GIS is essentially just making maps and understanding how different spatial trends affect things of interest. So later on, I'm going to talk about using GIS for measuring biodiversity. And like Claire mentioned, I am a student at the University of Wisconsin in Madison with the Nelson Institute. I'm finishing up my master's program in environmental conservation at the moment. And that program brought me to my partnership with the Urban Ecology Center, mapping that biodiversity, like I mentioned.
Great. So you've seen my thumbnail many times this summer and last summer, if you were joining for our Backyard Naturalist lectures then. But my name is Clare Eigenbrode, once again, and I'm the UBC's Science Communication Intern for this summer and the past summer. I'm very honored to be here. I graduated from Beloit College, just like an hour and a half south of here, here in Milwaukee in 2020 with a bachelor's in environmental studies with a focus on justice and citizenship and a major in Spanish and minor in journalism. And since then, I've become really interested in science communication through storytelling and science writing. So starting in just a couple of weeks, I'll be starting an MA in science writing at Johns Hopkins University, but I'll still be around, still be in Milwaukee. This is me repping some UBC swag underneath the Rainbow Arch last summer, and then this was just last week on a Slack call with my coworker, Paul the Snapping Turtle. I want to give some special shout outs this morning to Amanda, who is a mentor of Lindsey's on her project this summer, and with whom, along with Cameron, who's there in the other photo, I gave a very similar talk about biodiversity last week, beginning of last week, and I used a lot of their insights and ways of talking about biodiversity in this lecture. Also, just shout out to Felicia and Devin, who are two of our land steward interns this summer at Washington Park. So really grateful to have gotten to talk about biodiversity a little bit with these two coworkers last week, and to be able to carry it on now. So a little overview of what to expect. I'll be talking about just how we define biodiversity and how we measure it, and then a little bit about the history and theory behind the idea of biodiversity, and then I'll hand it off to Lindsey to talk about the work that she's done this summer. And we can hear a little bit more about how we can apply these ideas about biodiversity to our parks here in Milwaukee. So, because most of the focus of this talk is just one word, and it can be a little bit of nebulous word, if I just start with an Oxford English Dictionary definition with my own addition. So biodiversity is the variety, and I would add, and variability of life in the world or in a particular habitat or ecosystem. There's a few different ways to measure biodiversity. Most people think about it, they think of species diversity, which is the variety of different species within an ecosystem or within a geographic region or the world. And that's what I'm going to be focusing on today, because that is the focus of a lot of Lindsey's work. But you can zoom out and zoom in a little bit. So if we zoom out and look at ecosystem diversity, and that's within a given geographic area, the variety of different ecosystems. We'll be talking a bit about that. And then if you zoom in, we can look at genetic diversity, which is the variety of inherited traits within a species. So again, with this little graphic, you can see how we can kind of zoom in from this geographic region into one ecosystem and the species within it. Look at the species diversity within one ecosystem, like the boreal forest. And then from there, we can zoom in on one species and just look at the genetic diversity within one species. So starting with the big picture, ecosystem diversity is, as I said, the variety of different ecosystems within a given area. And ecosystem, we're on the same page, is physical environment and all of the organisms that interact with it. So that's both biotic and abiotic, living and non-living factors, features within an ecosystem are all part of the ecosystem. Rocks are certainly part of an ecosystem. Dead things like trees and animals, it's still biotic, but they're not living, are certainly still part of an ecosystem. They're still full of life. And of course, any ecosystem with humans in it, humans are part of that ecosystem. And then factors that we might not consider natural, man-made factors like buildings are also certainly part of an ecosystem if they're present and living things are interacting with them, which means that urban environments are certainly also ecosystems. That's why we have the Urban Ecology Center. And ecology is the study of ecosystems, so the study of the interactions between different factors within an ecosystem. And it's important to keep in mind, really anything that humans are studying, we're studying with ourselves in mind, right? So when we talk about ecosystem conservation, we're often talking about how to conserve ecosystems so that they continue to benefit humans. So ecosystem services are benefits provided to humans specifically by ecosystems in the natural environment, whether that's through recreation, whether that's aesthetic, or whether that's food or temperature regulation. So just kind of something that I like to keep in mind is that when we're talking about ecosystem conservation, we're often doing it with humans in mind, and how the ecosystem can benefit us specifically. So it's a very human-centric way of looking at the world. So zooming in a little bit to species diversity, which is what people tend to think of when we think of biodiversity. There's a couple of factors. The first one is just the raw number of species in a given area or ecosystem. We call that species richness. So that's just tallying up the number of species like within a group, or the total number of species within an ecosystem or in the world. So here we can see a map for mammal species richness worldwide. You can see that it's most concentrated in the Amazon, where there's up to 210 species of mammals. So this is just raw number of species, no other information. So it is missing a lot of information about how stable that ecosystem is. So there's a few ways to determine the stability of the ecosystem beyond just species richness, by looking at relative abundance of the species. So if we look at these two communities here, if we were to look at their species richness alone on paper, they would look identical, right? They both have 4 species of tree, but community 1 has a very even distribution of trees, and community 2 has the species A kind of has a monopoly. And so most land stewards would look at these two communities and say that community 1 is a more stable ecosystem. it's not it's not as likely to like it's not as difficult to sort of throw the balance off because we have more even balance of niches. So I'll let Lindsey talk about this a bit later, but Simpson's diversity index is one of the most common ways to calculate biodiversity or calculate relative abundance. So just keep that in mind when Lindsey's talking about her work. This is kind of a good visual to keep in mind for the higher relative abundance would be in community one, and even though these communities have identical species richness. And another aspect of species richness is alpha, beta, and gamma diversity. So alpha diversity is just the diversity of the community, just like the communities that we were just looking at. That was just alpha diversity, and then gamma diversity is diversity of a region, so that could be multiple ecosystems. And then beta diversity is the ratio between regional and local, or gamma and alpha diversity. And then one more aspect of species richness is functional diversity. So we're looking at the functional traits of different individuals within an ecosystem and grouping them together, for example, by diet, by size, or by sensitivity to environmental conditions. And then calculating the functional diversity by comparing it to the species density. So I think this is species density and then functional diversity of reef fish worldwide. And the reason that we use this is it acknowledges functional redundancy. That is, if like all species within a community had evolved to be the top of the food chain, then it would also not be a functional, or it would not be a very stable environment or ecosystem. So once again, we can't just look at species richness, and we can't just look at relative abundance, because they don't tell us the whole picture. They don't tell us they don't tell us whether like the community is really functioning the way that that the individuals within it evolved to. But there are a couple of snags with calculating functional diversity, because we're still learning a lot about different species and a lot about their niches. And so it's really only possible when we have a really clear understanding of most organisms' role within an ecosystem. So that's just one kind of pitfall to calculating functional diversity. And then zooming in one more time, genetic diversity. Genetic diversity is, once again, the variety of genetic inherited traits within a species. So maize is kind of a classic example of a species with really high genetic diversity. These are all ears of maize within the same species, but they have all these really unique varieties. And genetic diversity is produced by the same factors that lead to evolution. So mutation, which produces new alleles, genetic drift, which is a random change in the frequency of a certain allele within a population. Gene flow is the transmission of alleles from one population of the same species to another. And then natural selection, of course, is when resistant alleles, alleles that are resistant to a certain environmental change or certain environmental factor are the alleles that are most likely to be passed on. So it can often occur naturally, that is without human influence, but if we look at dog breeds, for example, dogs have really high genetic diversity because humans have bred them to. Humans, meanwhile, have very low genetic diversity comparatively. We're a really genetically homogeneous species. because we're a brand new species, relatively speaking, on the planet, and because there's a lot of passage of genetics from one population to another. And this varies greatly, like I said, from species to species. And it can be really beneficial to have high genetic diversity, because greater genetic diversity means that a species is more likely to have an adaptation that will help it be resistant to an environmental change and survive a change that could potentially lead to extinction if there was less genetic diversity. So one way to measure genetic diversity within a population is environmental DNA, or eDNA. If this really piques your interest and you want to Google it later, make sure you type environmental DNA, because if you just type eDNA, you'll get pictures of Edna Mode from The Incredibles, just a word to wise. Something that's really, so the way that we measure it is looking at cellular material that's been shed by multiple organisms within an environment. So we're often looking at waterways, and that could be like skin cells. We're often looking at feces. So it's often measured in water samples, and it's really cool because we can observe populations indirectly without actually seeing the species. We can even observe historical populations, but it's only really useful when we already have the genetic sequences of the different species that we're looking for. It's also really pricey, so that's the barrier to using eDNA to determine genetic diversity. So there's a few other places we can turn for biodiversity data. And the biggest one is community scientists. So this is a screenshot of all of the locations where community scientists who are community members who don't necessarily have formal scientific training, who are contributing to data collection. This is all of the locations that community scientists have reported data for the Monarch Larva Monitoring Project, which is a St. Paul-based organization that monitors monarch butterfly activity, including larvae and eggs and migration patterns. So it's amassed this huge database of community science data, which scientists can analyze to learn about migratory trends. So it would just not be possible without community scientists. I have so much respect and admiration and appreciation for community scientists who take their free time to record data like this for researchers, and they made Lindsey's project possible as well. So this is recorded in a few different ways. One is through volunteer groups, like the ones that we lead at the Urban Ecology Center, like our bird walks. And we are one of the, well, actually we're three of the locations that report data to the Monarch Joint Venture. Folks can also record data using apps like iNaturalist while they're out on a walk if they spot a species that they want to record in a database. And there are also folks who will just regularly check their backyard for species like with monarchs. So I want to talk a little bit about the history theory behind biodiversity. When I say we, I mean the sort of general public. And usually this question, when did we start thinking about or talking about a certain concept or idea, would be really hard to answer. But in this case, I have an answer for you. We started thinking about it on September 21st, 1986, when the National Forum on Biodiversity was held at the Smithsonian. This was like a huge event, like 14,000 attendees, lots of kind of ecology celebrity speakers, and the use of this word just exploded. Much like the song like a prayer. It really just became ubiquitous and just really captured the public imagination. Has captured mine. I know it's captured that of everyone on this call. There's something about the notion of biodiversity is really accessible, and it's become kind of a big phenomenon. So a few sort of key moments in the term and idea of biodiversity becoming so ubiquitous. It was coined very recently in 1980 as the term biological diversity by an ecologist named Thomas Lovejoy. And then that National Forum, which I mentioned, was held in 1986. That's when it really started to take off in the public. And then it was codified in 1992 with the United Nations Convention on Biological Diversity, which was ratified by 196 countries. So I want to talk a little bit about conversations about when biodiversity is useful and when it's the idea has drawn a little bit of criticism. So one of them is that there are pretty significant data biases in biodiversity data. So I actually know that this graph has been used in past Background Naturalist lectures. It's a pretty compelling one. We can see that charismatic megafauna like birds and mammals and flowers are really highly reported by community scientists. And then sort of creepy crawlies and creatures that are just a little harder to see and harder to observe are really underreported. So especially with community scientists who are kind of collecting this data as they go about their day, that's just a bias that occurs. And then a big question that Lindsey is going to address as well is whether community scientists are inadvertently collecting data on themselves. So we'll see, of course, higher community science data in higher populated areas. And we'll also see in areas with a higher median income, more community science data. And there's a lot of conjecture about why that is. I think, in my opinion, a lot of it is just higher income folks having more time to dedicate to collecting data, and also more resources to learn about how to collect data. And it's tough to extricate that phenomenon from the really well-documented effects of negative effects of environmental racism, environmental injustice, and redlining on biodiversity, which I would love to dedicate a whole lecture to in the near future. And then there's also a conversation about when it's useful to talk about biodiversity. So it can, because it's become this term that's really commonly used in the public, it can mean a lot of different things to different people. Surveys about how people conceive of biodiversity found that some people think of it as just a word for nature. So it's become this really broad and really common term. And then another recent move is to, among some ecologists, is to look at keystone species rather than just species richness, species abundance, and functional diversity, but to look at species that are an indicator of what we might call a healthy ecosystem. We saw with the reintroduction of wolves to Yellowstone that they completely transformed the health of the ecosystem. So that's a really common conversation right now among ecologists, is whether that's maybe a more useful way to measure the health of an ecosystem. And Indigenous American ecologists in particular are looking to incorporate Indigenous knowledge about keystone species into their work. And as far as when having high biodiversity is good, there are some instances on small islands with really sort of isolated ecosystems where higher species richness is really just an indication of high invasive species. And then another sort of controversial conversation is about whether we really should be working to save every endangered species, or if we should be focusing on keystone species, or if we should, if every species within an ecosystem really should be given the same weight. So that's certainly an ongoing debate. But that being said, I think that there is a lot of value to measuring biodiversity and talking about it and using the methods that we've been using, the way that Lindsey's been doing this summer. I think because species diversity is sort of understood by those outside of the scientific community, because it has really entered the public conversation and really captured the public imagination, like I said, that it can be useful because folks outside the scientific community are really used to hearing about biodiversity. And because we have so many similar measurements, it can compare really easily to those measurements. And also we just have this huge wealth of community science data on species richness and abundance. And I think I just love seeing what we can do with the analysis. And I just think that Lindsey's work is really cool. So I'm gonna hand it over to her now.
Okay. So like Claire mentioned, I focused my entire summer project on UEC and biodiversity and using GIS to kind of understand how different abiotic factors can affect biodiversity between the different branches. You can go to the next slide, Claire. So in terms of GIS, like I mentioned, it helps to visualize these spatial trends. And by this, I mean, you're able to kind of overlay the coordinates of different wildlife sightings over different things like land use that I looked at for my project, and create these maps that kind of inform decision makers on which areas may be more diverse and which areas may be less diverse and need some additional help in getting those biodiversity numbers up. And so for my master's project, I specifically looked at biodiversity, land use, and different aspects of census data surrounding each UEC branch. So in terms of species selection, I actually used a previous adaptive management plan that an environmental conservation student for my program made for UEC back in 2019, I believe. And some of the taxonomic classifications that UEC was interested in were squamata, which are snakes, odonata, which are dragonflies and damselflies, Lepidoptera, which are butterflies and moths, Apidae, which are bees, and then aves or aves. I'm still trying to find out how to actually pronounce that, which are just our birds. And then in terms of the spatial data, I used a couple different resources. So for biodiversity data, I used data that UEC provided to me. So data from wildlife surveys was really helpful. And then to supplement that, I also used wildlife sighting data from the Global Diversity Information Facility, or GBIF, which is this organization that seeks to allow the public to access wildlife data from around the world. So most of this data is actually uploaded by average people or community scientists who use different apps like iNaturalist or eBird to upload these coordinates for different wildlife that they encounter. And then for parcel data, I used the City of Milwaukee's GIS department, which was extremely detailed in categorizing land use for the hundreds of thousands of parcels in Milwaukee. And then for census data, I used the National Historic Geographic Information System, or NHGIS, which had a bunch of census data and polygon shapes that I could use in my GIS application for the year 2020. And so this slide here is just showing the map that I was able to create by placing the UEC parks on my map and then adding these red circles, which are 1 mile buffer zones, which I analyzed the different biotic or abiotic characteristics such as the land use and census data. And then these blue dots are the different wildlife sightings that I collected from UEC or GBIF. So you could already see that there's some difference in the number of wildlife sightings between each branch. So just going off of this map, you could see that the Riverside branch up in the northeast side appears to have more wildlife sightings compared to Washington Park and the Menominee Valley branch. And so in terms of parcel data, I was able to categorize the different parcels into 4 main groups. So the first one was the non-applicable parcels, and these parcels are essentially just parcels that are used for different utility services, such as sewage treatment plants in different sectors in energy. And then we have commercial parcels, then we have residential parcels, and then finally our public/quasi-public parcels, which you could consider our green spaces in this case, since this categorization includes things like parks, cemeteries, schools. So here is the same map that I showed before, but with the parcel data. So as you can see here, the different categorizations for parcels have different colors. So yellow is for commercial, non-applicable parcels are black, the public/quasi-public parcels are green, and then residential is purple. Then we also have a null group. These were just parcels that had land use codes that didn't fit into any of the categorizations, and that's just blue. And so then to make this easier, I was able to find the percentage of how much each parcel categorization took up in each buffer zone. So starting from Washington Park, you can see that majority of the buffer zone is made up of residential parcels. And then the Menominee Valley branch is majority commercial with 39%. And I think it's important to note that for this branch specifically, it's highly industrialized. And then for the Riverside Park branch, you can see that residential makes up the most of this buffer zone. And this buffer zone had lots of mixed use parcels. So things like condos over stores. And then for census data, I looked at 3 aspects of each UEC buffer zone. So I looked at the total population, the racial demographics, specifically the number of white, Black, Asian, and Latino identifying residents, and then also the income. And so this map is of the racial segregation in Milwaukee. So As you could see in the legend on the right, areas that are this kind of light blue denote areas where there is a higher number of white residents over non-white residents, which are just the other minority groups that I spoke of. And then areas that are more green show areas where there is a larger number of non-white residents versus white residents. So as you could see along the lake, there's a higher number of white residents as opposed to central and northwest Milwaukee, where it's predominantly not white. And then each buffer zone you could see, or you could get a better idea of the racial distribution between each buffer zone. So for Washington Park, it had a total population of 47,129 residents with a median income of $31,979. And this graph down here breaks down the racial demographics of the Washington Park buffer zone. So as you can see here, the predominant racial group is Black, followed by white, and then Asian and Latino. And then for Menominee Valley, it had a total population of 40,618 residents with a median income of $29,983. Again, this graph shows the racial distribution. So majority of residents identify as Latino, followed by white, and then Black, and Asian. And finally, Riverside Park had a total population of 55,396 residents with a median income of $47,639. And this branch appears to have the greatest difference between the number of white residents versus non-white residents. So as you can see here, white identifying residents are the predominant group in this buffer zone, followed by Black, Latino, and Asian. And so there were two different metrics that we went with in terms of measuring biodiversity between UEC branches. So like Claire mentioned before, species richness is just the number of species in an area. And then I also use the Simpson's diversity index. So the diversity index takes into account species richness and the abundance of each species in an area. And this value ranges from 0 to 1, with values closer to 1 representing greater diversity. And then over here on the right is a picture of that equation. So it gives much more weight to species that have higher numbers of occurrences as opposed to lower numbers. Oftentimes, I found that when working with this formula, it completely took out species that only had one occurrence. So that's something to keep in mind. And then actually looking at the biodiversity between UECs, we start with Washington Park. So it had a species richness of 209 and a Simpson's diversity index of 0.95, which is pretty high. And looking at the most abundant species in this area was actually the Canadian goose. And then for the Menominee Valley branch, it had a species richness of 199 with the Simpson's diversity index of 0.89, so a little bit lower than Washington Park. And the most abundant species in this area is actually the European starling, which I think it's important to note that this species of bird actually thrives in highly degraded areas, which makes sense for the Menominee Valley branch since this area is highly industrialized. And then finally for our Riverside Park branch, this had a species richness of 289 and a Simpson's diversity index of 0.97. So both the highest species richness and the Simpson's diversity index of all three branches. And the most abundant species for this branch was the American goldfinch, which tends to live in grasslands and prairies.
That's what we have. We can take some questions. Thank you for listening.